Okay, let's do section 3, the genetic code of life. Genes specify enzymes. A man named Archibald, which isn't that just the best name ever, described inborn errors of metabolism. Beadle and Tatum did experiments on a fungus, and they proposed that each gene specifies the production of one specific enzyme. Okay, so they hypothesized that one gene controls one enzyme. The flow of genetic information is from DNA to RNA to protein, which can be observed using our eyeballs or different methods. The genetic code of life, the mechanism of gene expression. Genes are found in DNA. DNA and genes specify information, but information is not structure or function. So it would be like the blueprints of a house. Okay, they tell you how to build the house, but they're not actually the house. Genetic information is then expressed into those structures and functions through protein synthesis, just like a house is built using the blueprints. It's basically the same process. The expression of genetic information into structure and function. DNA in a gene determines the sequence of nucleotides, which is transcribed into a molecule of RNA. RNA controls the primary structure of a protein. It determines the sequence of amino acids. So here we have a strand of DNA. Here's our sugar phosphate backbone, and here are our nitrogen bases. Remember, we start at the 5' end and we move toward the 3' end. Here's another picture of DNA, our sugar phosphate backbone with our nitrogen bases in the middle. It is then transcribed into messenger RNA. Okay, messenger RNA is very similar to DNA, but it is single-stranded and it uses a different sugar molecule. Okay, messenger RNA is divided into 3 nucleotide chunks, and each one is called a codon. Then that messenger RNA leaves the nucleus where it has been making a copy of the DNA. It moves to a ribosome where the ribosome reads the sequence of codons and it brings together amino acids to make the primary chain of the protein. RNA is a polymer. Remember, a polymer is a molecule that is made up of the same unit over and over and over again. It's made of RNA nucleotides. They contain a different sugar than DNA. They contain ribose sugar and 4 nucleotides. Very similar to DNA, but RNA does not use thymine. It uses a nitrogen base called uracil. So the 4 nucleotides are uracil, adenine, cytosine, and guanine. Uracil replaces thymine. There are 2 types of RNA. There's messenger RNA, which is a copy of one side of the DNA molecule. There is ribosomal RNA that, along with protein, make up a ribosome where proteins are then made. And transfer RNA, which is bonded or attached to an amino acid. And it helps transfer those amino acids to the ribosome so that the protein can be made. Here we have RNA. Okay, it's a single strand, but notice we have uracil instead of thymine, and our sugar base is a little bit different. Here's a table comparing the structure of DNA to RNA. In RNA, the sugar is ribose. In DNA, the sugar is deoxyribose. The bases in RNA are adenine, guanine, uracil, and cytosine, whereas the bases in DNA are adenine, guanine, thymine, and cytosine. Messenger RNA is single-stranded, whereas DNA is double-stranded. RNA does not form a helix, but DNA does. The genetic code. The unit of the genetic code consists of codons, each of which is a unique arrangement of symbols. Each of the 20 amino acids found in proteins is uniquely specified by one or more codons. The symbols used by the genetic code are the mRNA bases. They function as letters of the genetic alphabet. The genetic alphabet only has 4 letters, U, A, C, G. Codons in the genetic code are all 3 bases, or 3 symbols long. They function as words of genetic information. Permutations. There are 64 possible arrangements of the 4 symbols, taken 3 at a time. They are often referred to as triplets. The genetic language only has 64 different words. And those words each code for a different amino acid. So this is a table that is used to figure out what amino acid is being represented by each triplet. Okay, so you start with your first base over here. Okay, so then you move to the second base, which is up here. So if we have U, 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 we know that we're going to have phenylalanine because we go U, 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 and they all 3 line up here. Now, there is a wheel method that I find much easier to use. So you'll be getting a chart like this, and we'll go over how to use it, and a wheel that you'll be getting in class as well, and we'll go over how to use that. So properties of the genetic code. It's universal. With very few exceptions, all organisms use this genetic code the exact same way. They encode the same 20 amino acids with the exact same 24 triplets. Degenerate or redundant. There are 64 codons available for only 20 amino acids, which means most amino acids are encoded by 2 or more codons. There are no ambiguous, they're all unambiguous. There we go. Codons are exclusive. None of the codons code for 2 or more amino acids. Each codon specifies only 1 of the 20 possible. And it contains start and stop signals like punctuation. So the capital letter at the beginning of the sentence is a specific codon, and the period at the end of a sentence is a specific codon. And we're going to talk about what those are. So we'll talk about that in class. If you have any questions, let me know.